embryology. So the development of the nervous system for lecture one, we're only going to be talking about the brain. So let's do a quick revision for the brain as to how it's developed. Now, to understand the brain, there's two processes we need to know. First of all, the brain is essentially, and the spinal cord, the CNS, is going to be developed from a neural tube, all right? Something called as a neural tube. And essentially, the neural tube essentially looks like this, all right? And it will have two openings, essentially. And basically, from this neural tube, basically 1/3 of it is going to give you the brain, and the remaining 2/3, the remaining 2/3 is going to give you the spinal cord. All right, it's from this neural tube, all right, is going to be developing our nervous system. But before we even talk about the neural tube, we need to know how is it developed in the first place, right? This is what neurulation means, the process of formation of the neural tube. So let's take a quick gander at how it's developed, and then we could go to the second stage regarding the brain, that is. We're going to talk about the spinal cord, but regarding the brain specifically, known as vesiculation, because eventually 1/3 of it is going to form 3 vesicles, and then we'll continue from on. Tamam? Now let's start with neurulation. Let's translate some of this thing. So neurulation, like we said, is the formation of the neural tube, and it is induced by the notochord. خلي بالك, notochord and neural tube are not the fucking same thing. Notochord is a separate structure that is, if you look from this picture right here specifically, like before even that, do you remember the trigeminal disc? You remember ectoderm, mesoderm, endoderm? You do. Well, throughout development, and its process is complex, but just know that throughout development, the notochord is a layer of mesoderm that's going to be developed from the mesoderm, and is going to be located right underneath this ectoderm and right in between the mesoderm. All right, so the notochord is going to be located between the mesoderm and below the ectoderm. And phytata notochord is essentially, you see these little arrows coming out of it. The phytata in this notochord are going to release certain growth factors, all right, to the ectodermal layer above. Tamam? They're going to release growth factors to the ectodermal layer above. And what these growth factors do to the ectodermal cells is they're going to cause them to proliferate, all right? When does that happen? When the notochord is formed, all right? So all in all, the neurulation, which is going to be started by the notochord, is going to begin during which week? The 4th week. And when does it end? By the same week, by the end of the 4th week, all right? So let's look into what happens, right? What happens next is that when the notochord releases growth factors to the ectodermal layer above it, right? How does it help in the formation of the neural tube? Let's see. By beginning of the 4th week. So here it is right here. Now let's translate some of this work right here. So you see the notochord right here? You see this notochord? We say it was going to release growth factors to the ectodermal layer above. Well, what happens next is that these ectodermal layer cells are going to start to proliferate, and they're going to thicken. All right, what do I mean by thicken? Essentially, let's look at this. Let's imagine this is the ectodermal layer. All right. Now, let's imagine it's from about this size in diameter. All right. About this size in diameter. Well, after the notochord right here, let's say this is the notochord, it's releases some growth factors. They're going to increase in diameter. They're going to thicken further, all right? And when they thicken further, they're going to look like this. The neural plate is actually composed of 2 parts, like it's seen in this picture right here. Neural groove and neural fold. What does that mean? Essentially, what it's trying to say is that the more growth factors released from the notochord to the ectodermal layer, the more is going to be forming a structure known as a neural plate, and essentially parts of the neural plate are going to be proliferating on different sides. Essentially meaning what? It essentially means that basically that this lateral margin, all right, if we were to draw it right here, all right, what happened here? Basically, the lateral margins, this is the neural plate, the lateral margins of this lateral plate are going to start to elevate, all right? Basically, they're going to start to elevate from this side and this side. This side, these are going to go up. Meanwhile, the medial part right here are going to depress down, all right? So what is going to form is going to form 2 substructures, all right? The neural groove, the neural fold, the neural fold, and here the neural groove. Fold yani, get a folding, covering over, because they're going to come in both directions. We'll see. Neural groove, might not get a depression. Tamam? So the structure is going to look like this, essentially. Tamam? The structure is going to look like this. You see this groove right here, this depression going downwards? Tamam? That's your neural groove. And here is your neural folds on the lateral margins. Tamam? Like a hill. essentially. You take these together, you'll make up the neural plate. All right? Let's look at the picture below it. Like, you see it right here, right? One going like this, and this. And as we further develop more and more, you'll see, the neural groove gonna keep continuing. Basically, part of the ectodermal cells are gonna go down. Tamam? Keep going down from this neural groove. Part are gonna go up on the lateral margins from the neural folds. All right? So it's gonna look like this. It's basically gonna go from straight, tamam? From a straight line to essentially this. All right? Perfect. So together they make up your neural plate. Tamam? So what is the point of all this? What I'm trying to say is that basically the more growth factors are being released, these neural folds, all right, these neural folds right here, are going to come closer towards each other. They're going to come from this direction, and they're going to come from this direction. They're going to come towards each other. There's another picture. Let's see what happens. All right, there's another picture for it. We're going to see what happens next. So here it is right here. So first of all, we said before is that notochord, let's see, the notochord are going to release growth factors, and the ectodermal cell is going to take in to form the neural plate. Part of the lateral margins of the neural plate are going to elevate to form the neural folds, right? And then you got the neural groove, the pressure to the medial part, tamam. And then here's another cute thing to know about too as well. You see these cells right here? They're new additional cells coming out of the, kind of the edges of the neural folds. Well, these are called neural crest cells. Basically, some of the ectodermal cells are going to specialize as neural crest cells. And we don't have to know anything about it, but just some cute little thing to know is that when we form the neural tube, the neural crest cells are going to be basically being located between the ectodermal cells and the tube. They're going to be located in between. All right, I'm going to give many derivatives, but you don't have to know about it, so we're not going to focus too much. But anyway, from this picture, what we want to know is that basically the more proliferation can occur, the neural folds are going to come in contact with one another from this side and from this side, all right? And eventually, they're going to fuse with one another. The neural folds are going to fuse with one another and essentially detach itself from the ectodermal cell and form a tube, right? Which is known as the neural tube, all right? So this is the neural tube in its full glory, with the neural crest cells between that and that. And if you want to draw the notochord will be basically here, below it, tamam? Basically dorsal to it, all right? So there you go. We form our neural tube, essentially, tamam? So after formation of the neural tube, now eventually, another beautiful picture right here, the neural folds approaching each other, tamam. Fusion of these bad boys will convert the neural plate into the neural tube by the end of the fourth week, I should say, tamam, fourth week. And of course, the neural tube are going to separate itself from the ectodermal cell. And this is another beautiful picture right here. You can see the neural crest cells, right, between that and the ectodermal layer. Tamam. You can see the neural tube, you can see the notochord. But خلي بالك, the neural crest cells, like we said, they come from the lateral margins. Like you can imagine this is the light blue color, the neural crest cells. When the neural plate, the neural folds fuse with each other, the neural crest cells are going to separate itself. Tamam. Some of it are going to separate itself and basically be its own cells, essentially. That's what they're located in between. That's just another information you want to know, if you want to know what that is. All right, so we got the neural tube out of the way. We formed the neural tube. All right. Now, what happens, here's what I want you to basically imagine. You see this picture right here? This is the neural tube. Let's imagine we took this picture, all right? You see this is the neural tube. Let's imagine I brought my hand, I picked up the neural tube, and I dragged it out, all right? I dragged it literally out of the picture, all right? And then I flipped it basically vertically, all right? So from a Pringles can, a Pringles can horizontally became like this, vertically, like this picture right here, all right? So we're going to see we're going to see the neural tube in its full glory. And we can see that the neural tube in this picture is going to consist of two openings, right? On the cranial end, or cephalic end, you could say, and on the conal end. This is the cranial end, this is the cephalic end. On the cranial end, it has an opening known as the anterior neuropore, tamam? You could call it as cranial neuropore, same thing. And on the conal end, we got the posterior neuropore, which is also known as the choroidal neuropore. What we want to know is that by the end of neurulation, these two openings, the anterior and the posterior neuropores, should close at specific day of pregnancy. If they didn't close, then this will lead to anomalies. For example, they can lead to an anomaly in the spinal cord known as spina bifida. All right? Spina bifida. And if failure of closure of specifically of the cranial pore at 25th day, if that didn't close, then what happens is that there'll be no development of the brain. No brain either. And this is known as an anomaly called encephaly, as the encephaly. We'll see it in the end of the lecture, all right? But just know they're going to lead to many anomalies if there was failure of closure of these neural pores, all right? So the cranial neural pore occurs at which day? 25th day of pregnancy. The caudal neural pore is at which day does it close? At the 27th day of pregnancy, all right? So there you go. Now we mentioned at the beginning the neural tube. 1/3 of it is going to form the brain. The remaining 2/3 is going to form the spinal cord. For the rest of this lecture, we're going to be focusing on the 1/3 part of the neural tube. How does it develop? How does it basically dilate? Basically the cells in there proliferate to form the brain vesicles, and specifically 3 primary brain vesicles. We'll see. All right, we'll see. So we finished neurulation. We finally got the end result of neurulation, which is the neural tube. All right. Now, let's look at vesiculation, all right? Let's look at now the development of the brain known as vesiculation. Vesiculation might have vesicles. So if I'm going to talk about vesiculation, it's going to be consisting of 2 stages, all right? 2 substages, I want to consider it. Vesiculation. Basically, it's going to consist of something known as the cerebrovesical stage, the cerebrimary vesicle stage. And it's going to consist of the secondary 5 vesicle stage, all right? The primary vesicle stage occurred during the 1st week, and the secondary primary vesicle stage occurred during the 5th week. We'll see this in detail, but we're going to start with the first stage that occurs is the cerebrovesical stage, which occurs during the 4th week of pregnancy, all right? After neurulation, that is, all right? So let's see what it means by cerebrovesical stage. Cerebrovesical stage, and I like to further mention it's primary because here's the thing. Basically, this is the neural tube, all right? This is the neural tube. And basically what happens is that the neural tube, the cranial 1/3 of the neural tube, are going to dilate to form 3 distinct dilatations. And we're going to consider these as the primary brain vesicles. If there's primary, there's secondary. But for now, just consider these as the primary brain vesicles. What are these primary brain vesicles? Well, let us see. From anterior to posterior end. You got the first primary vesicle, we gave it a name, all right, known as the prosencephalon. The second, the second primary brain vesicle, the mesencephalon, all right, known as the midbrain. And then the final vesicle is known as the rhombencephalon, all right? How many vesicles did they form at the start? How many primary brain vesicles? 3. Proencephalon, mesencephalon, rhombencephalon, all right? This is actually what the neural tube looks like. It's going to be actually folded, all right? And it's going to form some flexures along the way. Flexures, some U-turns, essentially, like the cephalic flexure and the cervical flexure, all right? And there's another flexure known as pontine flexure. We'll see. We'll come back to this picture when we come at the end of the lecture, all right? But I just want you to know that if we were to draw it, basically the cranial 1/3 of the neural tube is going to form 3 distinct dilatations, SAQ, 3 primary brain vesicles, proencephalon, mesencephalon, and rhombencephalon, all right, from anterior to posterior end. Now that we got that out of the way, all right, what we next want to know is the second stage, known as the secondary 5 vesicle stage, all right? So you know about the first stage? Hello. 3 distinct dilatations? Hello. Now the second stage, basically during the 5th week, right after the first stage in the 4th week, in the 5th week we got the 5 vesicle stage, which is essentially basically mean what we will see. So essentially what happens, and we can draw it right here, what happens is that if we were to draw the neural tube, all right, let's take for example the proencephalon, all right, let's take the proencephalon real quick, all right, and let's have this on partial. We'll say this is the proencephalon. Right, we'll start with the first primary brain vesicle, all right? This is the proencephalon right here, all right? Now, this proencephalon is not going to give you additional vesicles. It's going to dilate to give you 2 parts, all right? It's going to be divided into 2 parts, all right? 2 secondary vesicles. Which proencephalon primary vesicle? Well, this bad boy is going to give you 2 or 3 in total vesicles. Let's see. So the proencephalon essentially is going to give you a secondary vesicle, tamam? And the secondary vesicle, let's say, the secondary vesicle, we will say, is known as the diencephalon. All right, the diencephalon. All right, this is a secondary vesicle. All right, so the proencephalon is going to dilate to be divided into two parts: the diencephalon and two additional vesicles. All right, secondary vesicles on each side, and you can guess what they are. In this side and in this side, it's going to be known as what, Nawaf? Basically, teleencephalon. All right, what is the meaning of teleencephalon? Just a minute. Teleencephalon. Teleencephalon basically means is going to form the cerebrum. So basically the proencephalon, when they further proliferate, is going to be divided into two parts, two secondary vesicles, all right? The diencephalon and the teleencephalon. The diencephalon, from its name, is going to give you the thalamus, right? It's going to give you the thalamus, it's going to give you the hypothalamus, the amygdala, and everything about this diencephalon, all right? The teleencephalon, there are two of them, all right? Why did I do two of them? Why are there two in each side? That's because the teleencephalon are going to give you the cerebrum. The cerebrum. والcerebrum عبارة عن two cerebral hemispheres. تمام? So it's going to give you two cerebral hemispheres, which essentially means it's going to give you, It's going to give you the frontal lobe, temporal lobe, on both sides, you know. That is the teleencephalon. So the proencephalon are going to give you two secondary vesicles. These are secondary vesicles. Secondary because they're developed from primary. All right? Two secondary vesicles. All right? In total, three vesicles. Three vesicles in total. All right? But two secondary vesicles from the proencephalon. All right? So that's basically is the proencephalon, what is going to develop. It's not going to give you additional. It's just going to divide into two parts. All right, and this is basically what it means: diencephalon and the teleencephalon laterally on each side. Tamam? Now, also, what's another cute thing about this whole thing too is essentially, and we have a picture for this too. Hold on a minute. We have a picture for the last point, and it's essentially right here. Tamam? Right here. So before we talk about that, you see, each vesicle will have a cavity. All right, each vesicle, like let's imagine. This is the teleencephalon, right? These two right here, and this is the diencephalon. In each vesicle, you see this empty space. These are lumens, right? These lumens, basically, as the structures surrounding it, like we'd say the thalamus right here, the cerebral hemisphere, the thalamus are going to develop, surround. Do you see these lumens right here? They're going to be developed into cavities, all right? And these cavities are very important because that's where the cerebrospinal fluid are going to be flowing throughout. And these cavities are going to be developing into ventricles, all right? What do we mean? Basically, you see the lumen of the diencephalon right here? Tamam? This lumen of the diencephalon will give you a cavity known as the third ventricle. We can see it even in this picture right here as well. Tamam? And the lumen of the teleencephalon, on the other hand, both lumens, what do you think they give? Remember from the anatomy of the brain topography, we saw in the anatomy of the basal ganglia, we saw a cavity on each side, tamam, known as the lateral ventricles. That's the one right here. It's going to give you on one side and both sides. So basically, one lateral ventricle right here and one lateral ventricle right here. And here is, of course, like we said, the third ventricle. And this is important because you see the thalamus is going to be sitting on the third ventricle, and whatever is going to be sitting on the lateral ventricles, the basal ganglia, is going to be the lateral ventricle, right? So that's what these lumens give. They're going to give you the ventricles. And basically, when they're developed, they're going to communicate with each other, all right? They're going to communicate with one another. But how? So a different structure, you don't have to know, but there's a different structure, how it works, that is. But through a different structure known as the interventricular foramen of Monro. Interventricular معناها between ventricles. Foramen of Monro is a scientist. So we can see what it means by that right here in this picture. You see this picture right here? You see this part right here? This is the third ventricle. All right, let's put it in the red color. This is the third ventricle, and this is the lateral ventricle. In between them, you see this? You see this connection right here? Do you see this connection right here? All right. This connection is known as the interventricular foramen of Monro. Basically, this allows for the lateral ventricle to communicate with the third ventricle. And the reason this is important is because of CSF. CSF can flow through this bad boy right here to the lateral ventricle to the third ventricle. All right, and then from the third ventricle, it can go to cerebral aqueduct and continue fourth ventricle and so forth. All right, that's the purpose of it. Basically, as a river flow. All right, that's the purpose of this entire thing. All right, so that's the point of this whole slide essentially right here. The proencephalon will differentiate into diencephalon, telencephalon. The lumen will give you one lumen of the diencephalon will give you the third ventricle, lumen of the telencephalon will give you two lateral ventricles, and the ventricles will communicate with another via interventricular foramen of Monroe. All right? All right, next we got the mesencephalon. We finished, by the way, the primary vein vesicles. What does it give? All right. Now let's go forward or from the back, the mesencephalon. Now the mesencephalon is very easy, very direct. Basically the mesencephalon right here is not going to give you any secondary vesicles. All right, we have 5 secondary vesicles. We looked at 3. The remaining 2 are not going to come from the mesencephalon. Instead, the mesencephalon itself is going to develop to form what? The midbrain. The midbrain, part of the brainstem. And you can already imagine what the next, rhobencephalon is going to develop next. But for now, just know as the mesencephalon is going to develop itself into the midbrain. And the mesencephalon, like any other vesicle, is going to have a cavity. It's going to have a lumen, all right? So this is the mesencephalic, the mesencephalon. This is its lumen, this empty space right here, all right? This is the mesencephalon and the lumen, the empty space. This lumen eventually, at some point, is going to become very narrow. And why does it become narrow? Because at some point during development, it's going to form what, Nawaf? The cerebral aqueduct. This is why. Do you remember the cerebral aqueduct? This is the cerebral aqueduct which was separating the ventral surface of the midbrain from the dorsal surface of the midbrain. And the cerebral aqueduct was very important because it was collecting CSF, all right? It was collecting basically CSF right here from the third ventricle and carry it to the next ventricle, which is the fourth ventricle, all right? The fourth ventricle. All right, so this is known as the cerebral aqueduct or aqueduct of Sylvius, the same thing. All right, so the mesencephalic lumen becomes very narrow and it's going to become known as the aqueduct sylvius, which connects the third with the fourth ventricle. All right, that's the point of this sentence right here. So we should know about it as well. So we finished the mesencephalon. Let's look at the rhombencephalon. Now for the rhombencephalon, this is going to give you the last 2 secondary vesicles. So basically the rhombencephalon, all right, this is the rhombencephalon. Or let's say this is the entire section of rhombencephalon. Remember what we said before. How many secondary vesicles? 5. All right. We looked at the first 3 on the prosencephalon. One basically diencephalon, 2 telencephalon. Now the remaining one's going to come from the rhombencephalon. So the rhombencephalon, the most posterior part, are going to divide into 2 parts. All right. It's going to consist of 2 parts. All right. So we're going to have one part on the anterior portion known as the metencephalon, all right? And the metencephalon, the meta, is going to give you or going to form what is known as the pons and the cerebellum. It's logic because if the mesencephalon from above is going to give you the midbrain, what's going to be developed from below? The remaining parts of the brainstem was cerebellum حواليها. So the metencephalon is going to give you the pons and let's say around the sides, the cerebellum, because the cerebellum is going to be from the back, all right? It's going to come from the back eventually, all right? So this is what the metencephalon, part of the rhombencephalon, going to give you the pons and the cerebellum. And the other posterior portion, the myelencephalon, on the other hand, the myelencephalon is going to give you what? If one going to give you the midbrain, if one going to give you the pons and cerebellum, what does the myelencephalon give you? Medulla. It's going to give you the medulla oblongata, all right? The medulla oblongata. So eventually, throughout development, the rhombencephalon is going to give you the pons and the cerebellum from the back. The myelencephalon is going to give you the medulla oblongata. Now, do you remember? There was a ventricle separating the dorsal surface of the pons and the dorsal surface of the medulla from the cerebellum. Do you remember from anatomy? What was this cavity called? Cherini was saying, this is the fourth ventricle. Tamam? So this is the cerebellum. You remember this cavity between the dorsal surface of the pons, the medulla, and the cerebellum? This is the first ventricle. And how's the fourth ventricle going to be developed? From the cavity of the rhombencephalon in general. So essentially in between the 2 portions, right here, in between them, all right? So we have the rhombencephalon. This is the lumen. It's gonna start developing. The cavity rhombencephalon will become known as the fourth ventricle. Essentially, when these structures, the pons, cerebellum, medulla, are gonna be developed, this cavity is gonna be given a different name, known as the fourth ventricle. All right, fourth ventricle, and we can see in the picture above it. All right, here it is, the fourth ventricle. Here is the cerebellum. Just imagine this, the pons and the or here it is, the pons and the medulla. In between them, the fourth ventricle. All right. Is going to develop from the cavity of rhombencephalon. Tamam? So this is basically everything about the three primary brain vesicles. All right? So let's review. Let's review everything by name. All right? We have three primary vein vesicles. Three primary brain vesicles, right? Let's see what they were. They were prosencephalon, mesencephalon, tamam, and the last one, what was that again? Rhombencephalon, rhomboid structure essentially. Rhombencephalon, tamam. The proencephalon is going to divide into two or three in total secondary vesicles. Going to give you two, telencephalon or two cerebral vesicles, tamam, telencephalon, and one diencephalon. The mesencephalon is not going to give you any secondary vesicles. It's going to form the midbrain. All right? So this is primary. Let's say this side is secondary vesicles. Now, the mesencephalon is going to develop as just the midbrain. Meanwhile, the rhombencephalon is going to give you the remaining, two vesicles to form the five vesicles in total. That's why they call it the five vesicle stage, mind you. The rhombencephalon is going to give you the metencephalon, and it's going to give you the myelencephalon. All right? This is literally all. How many vesicles did we count? Two, 1, 2, 3, 4, 5. 5 secondary vesicles, all right? And basically from 3 primary brain vesicles in total, all right? So this is the 5 vesicle stage. That's what you should write here, secondary 5 vesicle stage, all right? All this occurring during the 5th week, and it will end during the same week, which is the 5th week as well. Hello? Now, the next step is talking about the this is actually more relevant to the spinal cord, but this is talking about, okay, basically the telencephalon. Remember the cerebral hemispheres, right? The telencephalon right here. Basically, the telencephalon itself is going to be formed of 3 layers. Yani, 1 layer here, 1 layer here. So how many layers? 1, 2, 3. And here, 1, 2, 3. Yani, the telencephalon in each side is going to be formed of 3 layers, right? An inner layer, intermediate, and an outer layer. And if you remember from anatomy and even physiology, the cerebrum consists of 2 matters, right? A gray matter and a white matter, right? Gray and a white matter. And it's going to be consisting also of ventricles, which is going to be lined by, what do you think? Do you remember which were the cells that were lining the ventricles to secrete the cerebrospinal fluid? The ependymal cells. So you can imagine here, ependymal cells literally right here, right? Ependymal cells right here secreting cerebrospinal fluid into the ventricles. Because if you remember, the ventricles contain choroid plexus, right? Secreting, ependymal cells. So this is the point of the slide where basically we should know by the name. If you want to know anything else, we should know by their name. The wall of the telencephalon formed of 3 layers. The intermediate is the ependyma, meaning ependymal cells, right? We got the ependymal cells lining the cavity of the lateral ventricle. And this makes sense. The lateral ventricle, because what's ventricles in the telencephalon, in the cerebral hemispheres? The lateral ventricle, all right, on both sides, all right? The mantle layer next, forming the gray matter, contains nerve cells, and the marginal layer, which is the outer layer, which consists of nerve fibers, myelinated nerve fibers, which is going to be the white matter. Of course, we know the cerebrum, the outer part is gray matter, the inner part is white matter. Like I said, I think this is more related to spinal cord, but it's just to get an idea. It's the opposite, essentially, all right? But just know it as that for the exam, all right? We don't necessarily have to know everything about these things. Just know the 3 layers of the telencephalon. Enumerate the 3 layers forming the wall of telencephalon. There you go, all right? So it is about this point right here. And here's the last part, is the brain flexures. Now, this is the part we were at the beginning. Remember this picture? Remember when we were talking about the first stage, the 3 vesicle stage, how the cephalic end of the neural tube, all right? One side we're going to give you 3 distinct dilatations, the primary brain vesicles, where the tube itself, the neural tube, all right, it's not like a straight tube. Like we said, it's basically folded, all right? And when it's folded, it's going to give you the primary main vesicles, but it's also going to form three flexures. Remember flexures? Remember in GIT we took the flexures of the rectum and whatnot, right, this pathway? The same thing, but in the brain. And essentially, what we should know by their name, by the way, is that throughout development, the neural tube is going to grow rapidly, and it's going to bend itself forward. So let's say from like a straight tube, it's going to go from basically a folded tube. So it's going to look like a snake, essentially, all right? Like a snake. Like a snake, all right? And it's going to bend ventrally, and it's going to produce actually three flexures, all right? It's going to produce a third one later on, three flexures. And the three flexures, what are their names? The first one is known as the midbrain cephalic flexure. This is the midbrain cephalic flexure. Cephalic معناها cephalic is different from cervical. Cephalic معناها head, by the way. So cephalic flexure, right here, we have one flexure right here. The next flexure is known as the cervical flexure. The cervical flexure. Cervical معناها in the neck region. So this is the cervical flexure right here. And the third flexure, which is not in this picture, but the third flexure is known as the pontine flexure. Basically, where's this flexure going to be? You see the rhombencephalon? You see this rhombencephalon between the cervical and the cephalic flexure? Well, you see in this dorsal side right here, in the dorsal side, basically, as the rhombencephalon, basically going to give you many secondary vesicles, when it dilates, it's going to form a flexure known as the pontine. Pontine mana pons. So basically, it's going to form a flexure like this, tamam, and this flexure right here is going to be known as the pontine flexure from its name. Because you remember the rhombencephalon, right? Basically, the metencephalon, right, is going to give you the pons. So there's the pontine flexure. All right, it's not mentioned in this picture, but basically, on the dorsal portion, in an opposite dorsal direction, is going to give you the pontine flexure, all right, between the two flexures, all right? So it's going to produce the pontine flexure in between the two flexures. So 3 flexures in total. Enumerate the 3 brain flexures of the neural tube. There you go. Maybe an SAQ, maybe, all right? So those are the 3 flexures. Now, let's finally look at the congenital anomalies of the brain. It's very easy. We just got to know the causes and the features. So there's the encephaly, and معناها no, nothing, absence. Encephaly معناها brain, so no brain. And you can see in this child, he has no brain and basically no formation of the skull. And the reason why is, if you remember, of the neural tube, didn't it have 2 openings? The posterior and anterior cranial, or the cranial neural pore. Failure of closure in the 25th day will basically lead to encephaly. Because again, remember, there's the neural tube is actually exposed to the amniotic fluid from the outside. And amniotic fluid is urine. You don't want it. You don't want it to interfere with brain development. If it's not closed, all right, by basically with the help of folic acid, remember folic acid? Pregnant women had to take to prevent spina bifida. Folic acid is important for cell division. That's how they close the neural pores, by the way. That's how the neural pores even close in the first place. They take folic acid so the cells can divide, and basically, when they divide, they can cause closure of the neural pore, all right? Without it, deficiency or whatever reason will lead to encephaly, for example, all right? So failure of closure anterior neural pore lead to failure of complete development of the brain, skull, and the scalp, all right? He's gonna look like a homunculus, essentially. Important for OSPE. Now the next congenital anomaly is the microcephaly. The microcephaly, micro means small, something tiny. Cephaly means brain, means small brain. The reason why is there's a defect in brain development, all right? There's a defect in brain development, some fuck up in the brain development or whatnot. And the thing about the brain is that, the child, when an infant is born, you remember these skull fontanelles? Do you remember these skull fontanelles, right? Well, these skull fontanelles, what was their purpose? To permit easy growth of the brain. So the skull is originally not meant to be sutured completely, all right? It's not meant to be sutured. It's meant to be open with these fontanelles to allow for the brain to grow. And here's the thing, as the brain grows, so does the skull. The skull grows along with it, as of course when the fontanelles closes at the same time. So if the brain development, there's a defect, and this leads to development of the brain, this also leads to the cranium, the skull that is, to become small as well. And there's going to be the fontanelles, they're going to be there, but they're not going to be compressed, the brain's not going to grow, the cranium is still going to be the same size as it is, so the person is not just going to have small brain, he's going to have a small cranium as well, all right? So the circumference of the head, including the brain, will be smaller than normal. That's microcephaly. All right. Now, the last anomaly of the congenital anomalies of the brain, known as congenital hydrocephalus. Hydrocephalus can be acquired. We're looking at congenital hydrocephalus. And congenital hydrocephalus is characterized by an abnormal accumulation of CSF inside the brain cavities, essentially in the ventricles. All right, in the ventricular system, tamam? And the reason why is that for some reason, and this is very important, this is MCQ guaranteed, is because there is closure of the cerebral aqueduct. Basically what? Let's look at this picture right here. We can send this picture right here, all right? Let's get this picture here so we can explain it. So here you go. You remember the cerebral aqueduct? You remember it? You remember they was separating the midbrain dorsal surface, and it was connecting, and it was also connecting the third ventricle with the fourth ventricle, right? As part of a passageway. Well, let's imagine for whatever reason it could be, doesn't matter the reason, whatever reason it may be, the cerebral aqueduct, all right, the cerebral aqueduct, let's say it from this side right here, the cerebral aqueduct has obstruction, whatever reason, has stenosis, cerebral aqueduct stenosis, all right? هنا obstruction. What happens? The cerebrospinal fluid is going to try to come down, but it cannot. There's a blockage. And what happens is that the cerebrospinal fluid is going to go backwards. It's going to accumulate in the third ventricle, in the lateral ventricles. And basically, there's going to be accumulation of CSF in these ventricles. All right? There's going to be accumulation more and more, right? And this is going to be a big problem. Now, remember what I mentioned about the infant's skull? How it had fontanelle, so it's soft? Well, unfortunately, if there's accumulation of CSF, all right, if there's accumulation of CSF essentially, and we see the skull of the infant, basically the CSF can compress, it can basically compress the cranium with ease, all right? It's gonna basically push the cranium out. All right, basically the cerebrospinal fluid is going to compress the ventricles, the brain tissue. It's going to even compress the skull. The skull is soft, mind you. The fontanelles are not closed. It's not sutured completely, and that's why the baby will not suffer from just intracranial pressure. He's going to suffer from a huge head because the skull is very soft. So the skull can be very easily expanded by the cerebrospinal fluid because again, fontanelles is supposed to be soft for the growth of the brain, but instead, with cerebrospinal fluid accumulating, is going to be huge. That's why the baby looks like a fucking alien, all right? Due to, again, congenital or whatever reason may be, an obstruction of the cerebral aqueduct. هيجيك إلى MCQ, maybe a case to basically give you maybe diagnosis is congenital hydrocephalus, or he'll ask you for the cause. Congenital hydrocephalus is due to closure of the cerebral aqueduct or due to closure occurring where, cerebral aqueduct, where is it exactly? In the midbrain, right? Right here in the midbrain. So he could ask you as well that the cause of the congenital hydrocephalus could be due to closure of the cerebral aqueduct, or maybe he will ask you that the congenital hydrocephalus is due to obstruction where in the midbrain. All right, one of the two, either closed cerebral aqueduct or in the midbrain. Obstruction in the midbrain because the cerebral aqueduct is in the midbrain. All right, that's pretty much all that we need to know. All right, for the congenital hydrocephalus. All right. So that's pretty much it for the congenital anomalies, and that's pretty much it. Thank you for listening, I guess.